Welcome to the Explainer. Today, we are stepping into a microscopic world that feels, honestly, a lot like a high-stakes dating game. Except this one takes place entirely in the garden. We're going to dive right into the fascinating romance of two distinct rhododendron species, looking at how they connect, or, completely fail to connect on a cellular level. And as we'll see, mastering these intricate botanical secrets could actually pave the way for sustainable green technologies down the line. So let's jump right in. Now, you might be wondering, why is crossbreeding the perfect plant so incredibly difficult? Because horticulturists have this dream, right? To combine the absolute best traits of different plants into one perfect super bloom. You take the hardiness of one, mix it with the rare color of another, and boom, magic. Well, not exactly. It is literally so much harder than just rubbing some pollen together and hoping for the best. See, plants have spent millions of years building up these massive defenses, specific biological locks, and incredibly narrow windows of opportunity just to make sure they only mate with exactly the right partner. And that brings us to the ultimate invisible hurdle we call reproductive isolation. Basically, this is nature's way of keeping species distinct. It's a set of biological barriers that absolutely prevent different species from successfully mating and producing offspring. Sometimes these barriers are just about timing, sometimes they're geographical, and sometimes, as we're about to see, they are microscopic chemical roadblocks specifically designed to stop the wrong pollen dead in its tracks. Which brings us to Section 1. Meet the Botanical Matchmakers. Notice the sheer visual contrast between our two protagonists here. We've got Rhododendron latoucheae and Rhododendron molle. Now, both of these belong to the massive rhododendron genus, which is absolutely packed with over 1,000 species. But these two, they are from entirely different subgenera. Bringing them together is a major horticultural dream, simply because each one holds a crucial piece of the ultimate genetic puzzle. Let's take a look at their dating profiles, so to speak. Rhododendron latoucheae boasts these large, gorgeous, pinkish-white flowers. It has 10 stamens, and it is incredibly adaptable to its environment. Now, compare that to Rhododendron molle. It only has 5 stamens, but it features something highly coveted in the plant world, a striking, bright yellow flower, which is super rare in this genus. The stakes here are incredibly high, because if you can merge the strong environmental adaptability of latoucheae with the rare, brilliant yellow of molle, you've created an absolute masterpiece. But how exactly do you get them to match? That leads us right into Section 2, timing the perfect bloom. The window of opportunity we are working with here is incredibly brief. We're talking just 15 to 20 days. Through some really detailed field observations, researchers found that latoucheae starts blooming in early March, while molle starts a bit later, typically mid-March. This creates a highly specific, naturally overlapping window of just over 2 weeks where both flowers are open and actually ready for business. If they miss this window, well, the romance is completely over before it even begins. But of course, nature adds another layer of complexity. Both of these species are what we call protandrous, and all that means is that within a single flower, the male parts, the anthers that release the pollen, actually mature and do their job before the female parts, the stigmas, are even receptive to receiving it. It's a really clever mechanism to encourage cross-pollination with other flowers rather than just self-pollination. But man, it makes the timing of a matchmaker's job that much trickier. Let's zoom in a bit for Section 3, Microscopic Chemistry and Dormancy. Just look at these mind-blowing scanning electron microscope images. We're talking alien landscapes here. You've got the pollen grains, which look almost like these rough, compound tetrahedra held together by dense threads. Then you have the stigma's papile cells, these incredibly complex, textured surfaces that secrete mucus just to catch the pollen. This is where the actual magic has to happen. It is literally a completely different world down at this microscopic level. And the viability of that pollen, its actual power to fertilize, is a moving target. Latoucheae hits its absolute peak pollen viability of 96.23% right at the full bloom stage. But molle, it peaks earlier, hitting 89.33% right at the initial blooming stage, and then it immediately starts dropping off. So to make the perfect cross, you basically have to extract the pollen at exactly the right biological minute. But scientists actually discovered a fascinating workaround to this whole timing issue called short-term forced dormancy. See, when researchers stored the pollen at low temperatures, they noticed something incredible. The pollen viability would drop, hit a low ebb, and then miraculously recover its strength just a few days later. By chilling the pollen, horticulturists can essentially pause the biological clock, forcing the pollen to sleep until the exact moment the female stigma of the other plant is perfectly receptive. It's a real game changer. Which brings us to Section 4, a one-sided romance. So here is where things get really wild with this stunning asymmetry. When they used latoucheae as the mother plant and pollinated it with molle, it was a massive success. They achieved an 84.38% fruit set rate, producing really healthy seeds. But when they reversed it, using the yellow molle as the mother and latoucheae as the father, the fruit set was exactly 0%, a complete and total failure. This botanical romance, as it turns out, only works in one direction. So why on earth did the reverse fail so spectacularly? The microscopic culprit here is what we call a pre-fertilisation barrier. This is a biological roadblock where the mother plant literally stops the father's pollen tubes from traveling down into the ovary. It's a super aggressive biological defense mechanism, and understanding it requires looking at exactly what happens inside the plant's style. Watch the drama unfold at the cellular level here. When molle is the mother, the latoucheae pollen actually manages to land on the stigma and germinates. The tubes start to grow down the style and everything looks good. But then, the mother plant fights back. The microscopic channels inside the style begin forming what are called Kellos plugs. These plugs actively block the tubes. The pollen tubes get completely trapped, they start swelling, becoming disordered, and literally twisting in on themselves. Eventually, their growth is entirely arrested and the pollen tubes just dissolve away. Fertilization is completely denied. Bringing us to our final section, Section 5, Building Better Blooms. Okay, so we've solved the mystery of the unilateral sterility. But why are we looking so closely at this? Well, figuring out these plant biology secrets does so much more than just help us make pretty flowers. Plants are an absolute treasure trove of biological resources. By understanding how to break down these reproductive barriers and successfully integrate genetic structures from different plants, we can engineer entirely new resources. The source data points directly to how this basic research lays the biological foundation for some seriously cutting-edge technologies. We're talking eco-friendly functional materials derived from natural plant polysaccharides, green synthetic nanomaterials for environmental cleanup, clean energy tech, and even low-carbon circular building materials. Mastering the microscopic dating game of plants literally gives us the blueprints for a sustainable green future. It really makes you look at a garden completely differently, doesn't it? The next time you see two different flowers blooming next to each other, realize there is an epic, microscopic drama of chemical signals, Kellos plugs, and forced dormancies happening right in front of you. Which leaves us with this final thought. What other microscopic botanical mysteries, completely capable of unlocking our green tech future, are blooming completely unnoticed right in your own backyard? Thank you so much for joining me on this explainer. Keep wondering and keep exploring.